Michael Saad, which is a PhD student at Tufts University. He focuses on cultured meat and cellular aquaculture in the focus of his research. He's part of the Dr. David Kaplan's lab, which is engaged in a project about establishing continuous fish cell lines. The project aims to remove a bottleneck from cultivated fish food research and has been granted by Good Food Institute. Michael, please, you may share your screen.
Cool. So my name is Michael Saad, and I'll be talking today about perspectives on cellular aquaculture. So I just want to start out by thanking the organizers of this conference and thanking my colleague Andrew Stout, who actually made this PowerPoint slide template custom for our presentation that we gave earlier. And so a little bit about myself and my path to cellular aquaculture. So I actually did my BS at Rice University in Houston, Texas. And while at Rice, I discovered the field of cellular agriculture and ended up working at a company called Perfect Day Foods, which is a company in California that produces dairy proteins through genetic engineering and fermentation. And so after working at Perfect Day and working on milk and ice cream for 3 years, I decided to transition to meat and seafood. And so I've since joined the Kaplan lab at Tufts University. And so David Kaplan is the PI of the lab, and he has quite an extensive lab. And the cellular agriculture group shown in the middle is definitely his fastest growing team. And so this summer we had a number of undergrads working in the lab, and they actually made a logo for us. We're colloquially known as the meat team at Tufts University. And so they made a really cute little hamburger logo, which we've been sharing. And so the work in the Kaplan lab has primarily focused on bovine and insect cells. So you can see in the figure on the left, this is an image from a paper that Andrew Stout published, where he took bovine satellite cells and engineered them to produce carotenoids, so therapeutic meat to an extent. And then the picture on the right, Natalie Rubio and a new grad student, Sophie Letcher, have been working on insect cell culture for the purposes of cultured meat. And so this paper shown here was with Drosophila cells, and they've since moved to primary cell culture for insects. But now we're beginning to move towards seafood as well. And so similar to how we believe growing bovine or insect cells could be more environmentally friendly than cattle farming or other protein sources, we believe cellular aquaculture can have many of the same benefits over current aquaculture practices. And to give a rundown of the issues with some of the current aquaculture practices, we'll be talking about food security. So it's expected that by 2050, the demand for food will increase by 70%. And so currently fish support 20% of the global demand for animal protein. However, there are a couple of issues with fish as a protein source. One is that they have a relatively short shelf life and often have to be frozen in transport. And they also have a close to 50% byproduct. So while you mainly eat the fillets of the fish, a lot of cultures and a lot of people don't eat things like the head, the tail, the bones. And so that leads to a lot of waste. And so one thing that we look at is comparing marine and aquaculture. And you can see in the figure up here that marine capture has maintained relatively constant over the past few years, while aquaculture keeps increasing. But there's reason to believe that aquaculture could be approaching a plateau just due to overfishing, which we'll discuss in a bit. As far as safety, there are a number of foodborne diseases involved with fish and aquaculture. So you can see here a parasite in European anchovies. There's also been more evidence of antimicrobial drug resistance within aquaculture, and heavy metal contamination is also an issue, especially in terms of mercury, when you're looking at different waters. And in terms of the environment, many fisheries are actually being overfished. So that goes back to the issues with continuing to grow aquaculture. The number kind of varies depending on the source, but a conservative estimate seems like 30% of fisheries are being overfished, and that's only going to continue to increase as time goes along. Climate change has also been affecting fish populations. So there's global warming, there's been ocean acidification and drought that have affected and detrimentally affected a lot of the fisheries and a lot of the wild fish in the seas. And with the increase of microplastics in the seas, it's also reduced seafood spawning. So the figure here shows the biochemical oxygen demand for different processes. And you can see aquaculture has a really high biochemical oxygen demand, especially compared to some of the other food processes. And this is usually just due to the massive amounts of water that are needed for aquaculture. And so while these are some of the current issues with aquaculture, one thing that we are looking at is how cellular aquaculture can improve or provide an alternative to some of the current aquaculture practices. And so for cellular aquaculture, we believe in terms of food security that it'll be better in the sense that it can be a more flexible system. So I'm sure you're familiar with supply and demand. And whereas current aquaculture practices aren't super flexible, and so if there isn't demand for fish, but they're still growing the fish, those fish usually have to go to waste. With cellular aquaculture, we believe that we can fluctuate supply and demand. So if there isn't as high demand at a certain point, we believe that it would be possible to kind of scale down the operation. We also think that cellular aquaculture would remove the byproducts by manufacturing the target product only. So we'd be producing only muscle and fat, and to an extent, we wouldn't have a lot of the bone and the head and the tail waste that is seen with current aquaculture. In terms of safety, the facilities that would be producing this would be clean and sterilized equipment. So this is drawing a lot from chemical engineering and scale up in their manufacturing. So there would definitely be practices in place to ensure clean, sterilized equipment. There'd also be a robust testing of the products, both before and after fermentation. So you could do, after you clean, you can make sure that there's no contamination. And then obviously after the fermentation, you can confirm there was no unwanted growth in the culture. And there are we believe there will be fewer variables of production in the sense that we'll be starting with consistent sources of cells. And as opposed to cellular aquaculture, where you can have fish coming from all over the world, from different fishermen, from different processing plants, we believe that we would just have the cells that we need in a consistent production method. And then in terms of the environment, this is actually still to be determined. So there hasn't been a life cycle analysis, at least to my knowledge, specifically on cellular aquaculture. And while this is to be determined, a lot of this will depend on the culture parameters. So a lot of this will actually depend on how well the technology can progress moving forward. And so now that we've kind of talked about why we think there's a need for cellular aquaculture, we'll dive into what it actually is. And so similar to cellular agriculture, it's starting with cells, in our case, cells from fish or other seafood, and then adding a scaffold for those cells to adhere, and adding an optimized serum-free media in a bioreactor process to produce a piece of fish. So this will be usually involving two cell types, muscle and fat. And this is a slide from a presentation that Andrew gave a few months ago. But in terms of the inputs and outputs, we'll have stem cell isolations where we can take fish and get a primary cells from an isolation. We would then have optimized serum-free media, so that would include different growth factors, nutrients, and minerals, and then we'd scale those up in a bioprocess. And in terms of the outputs, we want to look at proliferation. So we want these cells to be able to grow as quickly as possible towards confluence, so we can look at growth curves. We'd also quantify differentiation. So in terms of muscle, that means we would quantify the amount of myosin heavy chain, be able to get a fusion index. And in terms of fat, we can start to look at lipid accumulation in different fat stains. And so while that's cellular aquaculture, there are two main reviews, at least to my knowledge, on cultured fish. So one of them is from the lab we're in. So Natalie Rubio and folks at New Harvest published a review titled Cell-Based Fish, a Novel Approach to Seafood Production, an Opportunity for Cellular Aquaculture. There's also a review from Greg Potter at Clean Research titled More Open Approach is Needed to Develop Cell-Based Fish Technology, starts with zebrafish. And so to my knowledge, again, these are the two big cultured fish reviews. And a lot of what I'll be talking about here comes from these reviews. And if I recommend, if anybody's interested in a deeper dive, these two papers are a great place to start. So these papers discuss the potential advantages of cultured fish. And so they have these considerations and potential advantage. So in terms of the oxygen requirements, fish are actually able to live in hypoxic conditions, so with less oxygen. And so the fact that they need less oxygen makes gas exchange less of a stringent requirement in the fermentation. And fish also have a high buffering capacity. So when you're thinking about pH and this high buffering capacity, a lot of times we see in fish cell culture, the bioreactor, the incubators don't require 5% CO2, which is a little bit different than mammalian. And so a lot of times when we culture our fish cells, we actually just add additional buffering to our media, and do them without additional carbon dioxide. In terms of temperature, fish can actually grow at a lower temperature in culture. So they can be as low, we see in like 13 degrees Celsius. For the most part, the cells that we culture are between 20 and 30 degrees Celsius. And in terms of karyotypic stability, they have a high karyotypic stability, meaning that they maintain their chromosome number doubling to doubling, and they're less variable in that sense. And then in terms of cell replication, there are studies that show that certain fish species are less prone to senescence than mammalian cells. Again, there are other species. This isn't true for all species, and some it might be the other way around, but it is promising that it looks like they're able to replicate for more doublings to a certain extent. And then while those are potential advantages, there are also some other considerations that are slightly unknown at this point about fish cell culture. So in terms of cell lines for cellular aquaculture, most of the cell lines that are available commercially are zebrafish cell lines. And so zebrafish are a really well-studied fish in different diseases. So the figure here is from a cancer research paper that used zebrafish. And while they're well-studied, definitely the most well-studied fish, they aren't edible or to an extent. And it's not because their tissue isn't edible, but they're really small fish. So they're only a few centimeters in length, which obviously doesn't make them too appealing from an edible perspective. For culture conditions, there are also different extracellular matrix proteins that could need to be optimized for fish cell culture. There are things like plate stiffnesses. So I think that a lot of the plates that you buy for cell culture are probably optimized towards mammalian cells, but maybe fish could be grown at a different stiffness of plate. You could also start to think about things like osmolarity and the venia, and kind of the amount of things that could be tinkered are endless. In terms of serum, a lot of actually all the fish cell lines that are available recommend using fetal bovine serum as a supplement. But of course, fetal bovine serum comes from cows and it's probably not optimized towards fish. And then in terms of the growth factors for growing fish cell culture, there's a low sequence identity to mammalian orthologs, which I'll discuss in a bit, but that could be another place for optimization to fish cell culture. And then in terms of antibodies, there are actually a few that are specifically available for fish, which could pose some problems down the line. And so to go over some of the fish cell lines, like I mentioned, zebrafish cell lines are the most common. And so here you can see two, ZF4 and ZEM2S. So these are embryonic fibroblast-like cell lines that are available. But again, zebrafish isn't a super well-known fish in terms of edible consumption. There are also Atlantic salmon kidney cells and European sea bass cells. So the European sea bass cell line was actually recently made available through the folks at Good Food Institute. So that these are available for purchase now. And it's definitely one of the more promising for cell ag in the sense that it's an edible fish that is well known. And then there are also other cell lines such as the common bluegill cell line or the rainbow trout gill cells, which are available through ATCC. And like I mentioned, all fish cell lines recommend fetal bovine serum or fetal calf serum as a supplement. However, as Cameron Semper, a postdoc at the University of Calgary and a New Harvest Fellow has noticed, there's actually poor sequence homology of fish and mammalian orthologs. So he noticed that at the highest it was 80%. So that's a good sign that they could be acting quite different, these growth factors. And so he's been working to produce fish-specific growth factors. And so the DNA gel that is shown here is from a New Harvest update that Cameron gave, where he's been able to produce the genes that will in turn produce fish-specific growth factors. And so that's definitely a very promising approach towards optimizing fish cell culture. And then in terms of antibodies, PAC7 and MF20 are two antibodies we use. So PAC7 is a paired box protein used for an early marker of myogenesis or myogenic differentiation, and MF20 we use for myosin heavy chain. So that's a later marker of myogenesis. And while these have been positively tested for zebrafish, they're untested for a lot of other fish species. So it's kind of unknown if these will work. And then when we look at the developmental studies hybridoma bank or DSHB, this is a University of Iowa collaboration where they are able to ship different antibodies. When you look at their host species, they're all mammalian host species. So they have mouse, rat, hamster, rabbit, and goat. And then when we look at the positive tested species reactivity, so this is, you can look up different animals and see if they've been tested for their different antibodies. Mouse, bovine, and chicken have 379, 48, 151 products that have been tested positively with their antibodies. However, when we query for fish, that number definitely drops. So for zebrafish, which I mentioned is the most well-studied fish, there's only 57 products that have been tested with positive reactivity. For mackerel, which is a fish of interest to us, there's only 2. And even if you just query for fish in general, you only get 74 products, which is obviously much less than, say, mouse, where it's 379 products. But the fact that there are all these unknowns mean that there's opportunities for research and opportunities for innovation. So things that we've begun thinking of exploring are things like co-culture systems. So there's a new PhD student at the Kaplan lab, Jake Marco, who's looking at algal co-culture with different fish cells and as an opportunity for improvement. We're also looking at different ways to immortalize. So I know that genetic modification isn't as encouraged in Europe, but we've been looking at ways to immortalize fish cell lines through genetic modifications, but we've also been looking at spontaneous immortalization through continued passaging. We could also begin to explore transdifferentiation. So it would be switching between like myogenic and adaptogenic differentiation. And we could look at metabolic engineering. So there's an idea from Andrew's paper on bovine cells where he got these cow cells to produce beta carotene by genetically modifying them to produce enzymes that would eventually lead to beta carotene production. So similar metabolic engineering projects could be explored either with or without genetic modification. And in terms of potential applications of cellular aquaculture, so the one that obviously comes to mind is it's cultured seafood for edible purposes. And these could either be hybrid or whole cut. So if you're thinking about hybrid product, fish fat is really valuable for, or really desired for its health benefits. So you could think about a hybrid product with fish adipocytes and plant-based protein. Fish feed is also really popular. So that could be another area of exploration for cellular aquaculture. And then fish oils are seen at a lot of drugstore counters here in the US for their health benefits. And you could also begin to look at therapeutic foods. So combining that therapeutic food and metabolic engineering idea with fish oils, one idea that's come to mind is that omega-3 fatty acids are a really touted health benefit of eating fish. And so through metabolic engineering, you could start to look at different enzymes and pathways that could increase the flux towards those omega-3 fatty acids. And then you could include those in a product like a fish oil, which is touted for its health benefits. And so there are a number of companies looking at producing cultured fish on the market. There hasn't been a cultured fish product available yet, but there are a number of companies who seem close. So there's a company called Wild Type who's producing cultured salmon. Finless Foods produces cultured tuna and have been transitioning and exploring plant-based tuna as well. Blue Nalu has been looking at yellowtail and shock meats, in Singapore have been looking at crustaceans and specifically shrimp. But we also think there's a need for academic research. So these are a couple images from our lab where we've been looking at mackerel. We've been looking at their ability of our primary cell culture. So we did a cell isolation of mackerel and we're looking at the ability for those to be differentiated into different cell types. So like I mentioned, we used MF20 or a myosin heavy chain and actually did have a positive reactivity with the mackerel cells that we have. So it was a definite positive sign for its fish cell culture. And so we're able to stain myosin heavy chain and mackerel and show myogenic differentiation, myofibers. And we've also been looking at adipogenesis. So we looked at lipid accumulation where we provided adipogenic media to those same cells. And it's a little harder to see. We don't have staining yet, but we do think that we're starting to see some lipid accumulation with little droplets that are forming within these cells. So like I said, we do think there's a need for academic research, and the Kaplan Lab is interested in any questions or advice that anyone has for fish cell culture or any potential collaborations that people are interested in. And so that's the wrap of my presentation. Here are some references from a lot of the images that I used. And I want to thank everyone for their time. I provided my email here if anybody would like to reach out, and I'll open the floor to questions.
Thank you so much, Michael, for such a nice presentation. Now, everyone, please raise your hands or write in the chat for questions. So we have a question from the chat. What other fish would be good to target for cell line isolation and establishment?
Yeah, so that's a good question. I think it's up to the researcher. That paper that from Greg Potter and Clean Meat promoted zebrafish as a good cell line to explore, just because it's so well studied and it's a lean fish, meaning that you wouldn't have to focus as much on the fat. You could focus more on the muscle. But that being said, we've tried exploring different edible species like salmon to varying success. But I think there are benefits to different fish. So if you want to start to look at fat a little bit more, those fatty fish have a higher fat ratio and more opportunity for improvement on the adipogenic side. But again, lean fish could be an opportunity if you want to focus more on muscle.
Another question from the chat. What measures do you think would have to be implemented to ensure that the cell line does not change over time?
Yeah, so that's a good question, and definitely something we're starting to think about. I think consistency in the culture is definitely key. So consistent media, consistent feedings, consistent passagings. But in terms of kind of checking whether or not the cell lines are changing, it seems like karyotyping. So looking at the chromosome number is definitely a measure that's usually needed when developing a cell line. So I think karyotyping would be most recommended just to ensure there are no changes. As far as preventing changes, that's something that we're still looking at exploring.
Another question from the chat. Is it harder to make culture marine products or culture meat?
So that's a good question and something that it's unknown. So that's why we're doing this research. Like I mentioned, there are potential benefits of the aquaculture, cellular aquaculture, like the pH considerations and the hypoxia and things like that. But those haven't been as well studied at the cell level. And so that's where the research we're doing, and hopefully the research other groups are doing in cellular aquaculture, can start to fill in some of those gaps and start to answer some of those questions.
Another question from Bernard. I'm a bit curious about the aspect of omega-3 of cultured fish. Naturally, the omega-3 of fish come from the diet of fish. What strategy might be utilized to ensure that cultured fish contain omega-3 as conventional meat?
Yeah, so that's definitely a good consideration. It seems like a lot of the the omega-3 fatty acids, again, come from that diet. But we think that by providing some of the precursors to that and providing the enzymes for the fish cells to actually convert those precursors into the omega-3s, that could be an alternate strategy. So obviously one strategy could be to include the omega-3 fatty acids in the culture media and have those lipids be accumulated that way. But we're also interested in exploring like enzymatic processes in terms of promoting flux towards cultured omega-3 fatty acids.
Another question from the chat. Based on your fat research, I was wondering, do you know if there are major differences between visceral and intermuscular adipocytes?
So that's a really good question, and it's something that to this point, these cells that we have are rather new, and we haven't done tons of characterization, but there are questions like that we're going to have to begin to explore. And so one thing that we're interested in doing is kind of separating the muscle cells that we have and the cells that can produce fat that we have, and be able to explore them and study them more in depth and kind of more focused on the one cell type. Because right now, it seems like in the cells that we have both cells that can form muscle and cells that can accumulate lipids, but we haven't really separated the two. And that's definitely something that we're interested in studying moving forward.
So apparently someone is interested in cultivated fish food. There is a question. Are there any PhD positions available on this?
I would say definitely feel free to reach out to me. The team at Tufts and the cellular agriculture team is growing, and there's been a lot of interest in a lot of the cellular aquaculture work. So if you're interested in learning more about the lab and learning more about the research we're doing and joining the team, definitely reach out to me and we can put you guys in touch with David Kaplan, the PI, and the rest of the team.
Another question. What marine product do you think is the most important to target with cellular aquaculture? Something like bluefish tuna or other fish products that are accessible to more?
Yeah, so bluefin tuna is definitely one that gets brought up a lot just because of the concerns over the overfishing of these fish and the really high price. So I think in our minds, that's something that we'd like to explore. Getting access to bluefin tuna, especially like fresh tissue, is definitely a bit difficult just because they aren't really farmed and they're more wild, and these fish are gigantic. So they can be on the order of hundreds of kilograms. But I think that is a really interesting species to explore, and it's one that we're definitely interested in as well.
Another question from the chat. You're trying to isolate and establish cell lines from Atlantic salmon. Was the fish choice random, or is there something that renders salmon the better choice?
Yeah, so we actually got a grant from the Good Food Institute. It was actually before I joined the lab, so I kind of inherited the grant. But I think the rationale for Atlantic salmon is, again, it's a fish that's well known and that has, especially in the US and I'm sure in Europe, is probably the most popular fish choice for consumption. So I think just given its popularity and given the demand for salmon, I think that was the motivation for that choice.
Another question. iGEM Uppsala is attempting to optimize the serum-free media for meat. How is the serum-free media for fish?
Yeah, so it's something that we haven't yet explored. So we're working more on characterizing cells and isolations and growing them with serum. But there are groups like the Uvicipore lab at Virginia Tech, who has begun looking at serum-free media. And so they have a YouTube talk from the Good Food Institute, Reza Uvicipore, professor, where they talk about some of their approaches to serum-free media. It's definitely been less explored than mammalian cells, but that just means there's more opportunity for people to fit in and do that research.
Thank you so much, Michael, for your presentation.